Okay. Okay, guys. This is the place where yesterday actually we stopped, if you remember. We started with what is a marker, and then we decided that we are going to talk about anonymous markers and also what candidate gene markers. And I defined what anonymous markers are and what candidate gene markers are. All right? Okay. Since we stopped here yesterday, if you remember, I was talking about mini satellite being a multi-locus, multi-allelic marker. Now, I was just starting with microsatellite, and microsatellite is now termed as single locus, multi-allelic. Single locus, multi-allelic. Now, when you're talking about single locus, multi-allelic, you'll have to understand why is it single locus, multi-allelic. If you remember, this is also a tandem repeat. We talked about this. This is also a tandem repeat of 1 to 6 base pair, right? Repeating one after the other. And the major differences between the individuals would be the number of repeats. That's what I told you. And here again, the main reason for the difference in the number of repeats is mainly because of DNA slippage again, right? Now, these microsatellites, however, are detected by means of primers SID, right? Primers are actually adjacent to these repeat regions that are tandem one after the other. And these primers are very specific. For example, you have, as per the FAO, if you are talking about FAO, as per the FAO, you have around now 30 primers, right, to characterize cattle breeds. And similarly, you have certain set of primers of microsatellite primers I'm talking about to characterize cattle breeds. So that's how FAO was initially defined. Now that NGS is in vogue, right, no one is characterizing by using microsatellites. But earlier, like for cattle breeds, like for sheep breeds, for even like goat breeds, there are a defined set of microsatellite primers I'm talking about. So when you're using primers, what's happening? You are targeting a single locus. You are targeting a single locus. And if this is a single locus, now what's happening? At this particular single locus, if you are amplifying, now let us say you run a gel, like what we have done yesterday, individual 1 and individual 2. Let's say this is individual 1 and this is individual 2. Now, for a set of primers, if you are running a gel after amplification, let's say you'll get specific bands here. In one particular individual, again, we are having around 20 repeats. And this 20 repeats, let us say, will account to around 120 base pairs, because the maximum is 6 base pairs. So it's around 120 base pairs, right? Now, in the second individual, wherein you have run the same set of primer, right? If it has only, let us say, 14 repeats. So 20 repeats, if you have looked into as 6, and let us say 14 repeats, if you are multiplying this, it is 6 4s are 24 and 2 and 84 base pairs. So you will have an 84 base pair fragment in the individual 2. In individual 1, you have around 120. And once again, this is very important, right? This is again a codominant marker. Codominant marker. Now, two things we have defined. One, it is a single locus. The other one is a codominant marker. Now, what did I tell you? I also told you it is multi-allelic. Why is it multi-allelic? Because in individual 3 per se, there are 15 repeats. Then it becomes 90 base pairs. You understand? I'm taking a maximum size of the microsatellite for you to understand. If this is 84 and you will be having a 90 somewhere here, this corresponds to 90 in individual 3 and this corresponds to 84 in individual 2. So likewise, at the same locus, you are having multiple alleles. There is a possibility of detecting multiple alleles in a population. Having said that, you'll have to also understand about codominance. Per se, in the same individual, right? Here, we have taken that in both the individuals, it is similar as 20 and 20 base pairs on both the homologous chromosomes in all the individuals. Now, per se, in individual 4, if you are talking about, in individual 4, you have around, let us say, on one maternal chromosome, you have around 20 repeats. And on the other paternal chromosome, you have around 15 repeats. Now look what happens. Now what are we looking at? We are now looking at the 4th individual, right? So at the 4th individual, if you are looking at, you just extend the gel to the 4th individual. This is the 4th individual. Let us say you have around 4 individuals now. And if you are looking at the 4th individual here, right? Now, what did I tell you? This corresponds to what? 90 base pairs. Here, since this is 15, you'll have a 90 base pair fragment here, as well as what? You will also be having a 120 base pair fragment. It means like both of them that are present on both the chromosomes. That's what I told you. Whenever you're referring to codominance, you are just talking about the presence of the alleles on both the homologous chromosomes. Homologous chromosomes. This is very important. Codominance, if you are referring to identification of alleles on both the homologous chromosomes, maternal and the paternal. Right. That is regarding the microsatellites. Now we are left with one last marker that is called as AFLP. AFLP. AFLP is what? Augmented fragment length polymorphism. That this should be very clear. Augmented fragment length polymorphism. Okay. Now, if you are looking at, this is again an anonymous marker. We'll first look at the technique, how it goes by, and then it will be easy for us to understand. Right? Wherever there is a word fragment, now you clearly understand, it means like it has to be broken down into pieces. That's what you understand, right? So now that you understand that fragment involves restriction enzymes, we'll take this way. This involves restriction enzymes, right? That is what we are talking about. Okay. First, what do we do in AFLP? AFLP involves, like, let us say you have DNA, you take this DNA, cut this DNA with a frequent cutter first and a normal cutter next. Means like you cut it with a restriction enzyme that has a restriction recognition site of 4 base pair and as well as 6 base pair, right? So frequent cutter and a normal cutter. Normal 6 base pair cutter. So now look what's happening here. Now what do you have here? As you proceed, when you cut it, this particular fragment that you have, that the fragments of DNA that you have here on one side will be having a 4 cutter recognition site as well as a 6 cutter recognition site. That is the place where, yes, you do have fragments with 4 cutter on both the sides, right? But I'll tell you how they'll be removed in the next steps. If at all you have a 4 cutter, there is a possibility that you have a 4 cutter here and a 4 cutter site on both the sides. You have a 6 cutter here and a 6 cutter here on both the sides. There is a possibility. But only these fragments are selected. How are they selected? Because now let us say, like, you have fragment, right? this is a fragment. I'm just drawing a fragment. Now this is the overhang, which is actually occurring after the initial cut of 4 base pairs. Means you cut it together, but anyways, and this is the overhang that you get after you cut it with the 6 base pair cutter, right? Now what happens? You attach adapters to both the sides, which are complementary to these sides. Now what happens when you attach adapters? Adapters will go and bind here, this place, and adapters will go and bind here. Adapters are what? Adapters are double-stranded molecules which have complementarity with these regions that are left as overhangs on both the sides. Now what's happening? When the adapters are bound here, now in this particular selection, right, there is always a chance again. What's happening? This 4 base pair adapters will be bound to 4 base pair on both the sides. 6 base pair adapters, 1 6 base pair here and 1 4 base pair here. Whereas 6 base pair adapters can go and bind at these places. There is a possibility, I'm just telling. Fine. Still, the probability of eliminating these fragments has not occurred yet. So initially, what are you doing? You are taking the DNA, cutting that into pieces. How are you cutting? You are using a 4 base pair cutter as well as a 6 base pair cutter. So you have an overhang on one side, 6 base pair, on the other side, 4 base pair. You also have fragments having 6 base pair on one side and 6 base pair on the other side, 4 base pair on one side and 4 base pair on the other side. Then what are you doing? You are attaching the adapters. This step is what? Attaching the adapters. The adapters. Now you have how many kinds of fragments after attachment of adapters? You have a fragment with ideally what you would need, 4 base pair on one side and the 6 base pair on the other side. You do have fragments with adapters with 4 base pair and 4 base pair on the other side. Now, in the next step, look what happens. You target primers. You already know. For example, now what happens? You have so many fragments. I'm just showing you one fragment. You have a second fragment here, right? You have a second fragment here. And this second fragment again has a 4 base pair here and also a 6 base pair here, right? And you may also have some other fragment, a third fragment here. And all these fragments are different, right? Don't assume that this is fragment 1, this is fragment 2, and this is fragment 3 here. And all these fragments are different. So you have, again, you have what? You have a 6 base pair fragment here, and you also have a 4 base pair fragment here, like 6 base pair and 4 base pair. Now what is happening? Just have a look. See, look at the technique now. Initially, you had DNA, you cut that with restriction enzymes, and then attach your adapters. Now target your adapters with primers. Target adapters with primers. With primers that are specific to your 4 base pair and 6 base pair. That are specific to your 4 base pair cutter and your 6 base pair cutter, right? Now what is happening? Just have a look. These primers being generic primers means they can bind to this particular region, right? And also bind to this particular region, right? And as well bind to this particular region and also to this particular region, and also bind to this particular region and also bind to this particular region. So in one single go, you are using the same set of primers to amplify all different fragments. You understand? That is the catch here. By amplifying all these different fragments, what are you identifying? You are identifying the difference in the individuals across. This is in one particular individual, right? This, when you are looking to looking at is only in one individual. In the second individual, again, you have a different set of fragments, and the same procedure follows. You are attaching. Now, having said that, now your primers are there. And what are you doing? You are amplifying this. You're doing an amplification, and after amplification, you are trying to identify the polymorphism. Now, the catch here is what and how the polymorphism occurs. See, how the polymorphism occurs, you should understand. Now, let us say, what is the first reason for polymorphism? The first reasons for polymorphism in AFLP. Reasons for polymorphism. Right? What are they? Okay, the first one, what is that? The first one is presence or absence of the restriction site. There may be a difference in the restriction site, presence or absence of RE. Right? That's always there. Right. Next, the second one. See, if the RE site is there in one particular individual at this particular point in cutting this particular fragment, there may be a mutation at that region resulting in that particular fragment not being present or not cut in one individual and may cut in the other individual. Okay, and what next? The first one is presence or absence. Next, what you should understand is, this is the next one. Look at this. When you have a fragment, we'll use the same colors so that you understand. When you have a fragment, this is the fragment I told you, right? This fragment, the difference between the individuals within a fragment can be because of a deletion or an insertion. So if you are amplifying this particular fragment and you are expecting this fragment to be, let us assume that this fragment should be around 500 base pairs, right? That is what you are assuming, right? A particular fragment. And this fragment, if at all it is there and the cut site is as well there in the second individual, you expect that it should be 500 base pairs. But sometimes it may so happen within the 500 base pairs of that second individual, there may be a deletion or a insertion, resulting in the size of that particular fragment not being 500 and decreasing if it is a deletion and increasing if it is an insertion. You understand? So, what is the second reason for polymorphism? Insertion or deletion within the fragment. And the third reason is presence of mini-satellite or microsatellite within the region. You understand? Mini-satellite or microsatellite, it always depends upon the number of tandem repeats, mini-satellite or microsatellite repeats within the fragment. This fragment of 500 base pairs, if it is expected in both the individuals, if you have a mini-satellite or microsatellite located within that particular fragment, there is every possibility that the number of repeats of the mini-satellite or the microsatellite may be different between the two individuals, resulting in the difference in the size of that particular fragment being amplified from an individual. Okay. Now, having said that, three particular reasons are very clear. There's one more fourth one, right? And what is this fourth one? See, while designing the primers, people have designed primers, right? For example, this is the fragment. This all of you should understand. Now you have this adapter sequence, right? Now you are targeting with a primer. Now this primer, this particular primer, this inset, I'm just magnifying, right? If this is the primer, people have designed primers. I told you, this part will be completely complementary to that of your RE enzyme. site, RE site, and therefore it is going and binding to that particular adapter. That is very clear. Now, what people have done is, in addition to that, they have added a random triplet at the 3' end of the primer. This is the 5' end, and this is the 3' end. There is a random triplet that is added. Because what happens when so many fragments are cut and so many fragments are being amplified, there is always a possibility of getting a smear after the amplification. Because this will amplify, the other one will amplify, and this will amplify. So what people thought is like we should reduce the background noise of these fragments, and we'll incorporate 3 random triplets into the primers. Because of this incorporation, the number of bands that are amplified will be reduced, right? And depending on the variation of these triplets in these primers, there is a possibility that whether the primer would exactly bind to that particular adapter or not for amplification, and thereby reducing the noise, in the sense like what happens, you will not be getting a smear. Or otherwise, when once cut with the restriction enzyme and you're using your adapters and amplifying, you'll be having so many fragments. When you run it on the gel, there is every possibility that you will be getting a smear. To reduce that, people have used a triplet, and thereby reducing the probability of the primer going and binding to almost all the fragments. And because of this random triplet, they'll be not binding to your adapters sometimes. And those that are binding, right, will amplify. So what is this? The variation in the, this last one is what? The variation in the random. Variation in the random. Variation in the random triplet that is there, right? So now let us see how we'll focus on 2 individuals. We'll take the DNA in 2 individuals, and then we'll focus on how this occurs in 2 individuals, right? We'll just see. You have a DNA, right? And this DNA is now cut. This is individual 1, let us say. This is individual 1. Now, this is individual 2. We'll take this. This is individual 2. This is individual 2. I'm just writing IND2, right? Now, what is happening here? Look at this. So, if you are looking at this, right, individual 1 and individual 2, you are initially using what? Set of restriction enzymes, a 4-base pair cutter and a 6-base pair cutter. That's what you have used. Same thing here. You are using a 4-base pair cutter and a 6-base pair cutter. That's what you use, right? So you are amplifying this in the second individual. Now what happens? There will be several fragments that are cut, right? I'm just drawing a fragment. They're all double-stranded molecules. Don't get confused saying that sir has drawn a single strand here. They're all double-stranded. For ease, I'm just drawing for different fragments, right? Now there will be different fragments that are there here as well. We'll be having different fragments here. Now what happens? The next step is what I told you. Attach the adapters. Attach adapters. Now what do? After doing this, you attach adapters on both the things for all the fragments here that you have, right? Adapters are one and the same, right? So I'll draw with the same color. You'll be having adapters on both the sides, right? And you'll also be having adapters on both the sides here, right? This one here, this one here, and this one here. This one here, and this one here. And similarly, you'll be having a fragment here, and also you'll be having a adapter on both the sides, right? Likewise, you'll be having adapters here. We'll just draw the adapters quickly. Okay, you'll be having adapters here. Now what happens here? You understand why there is difference. You'll amplify. Next step is what? You will simply amplify. You will simply amplify. Now, once you amplify, what happens? The primers would bind to these fragments on both the sides, right? Primers would bind to these fragments on both the sides. That's what happens, right? These primers would bind and would amplify to your these things. You have it here, 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 and you have it here. Now what happens? Your primers would go and bind here, to this place, and to this place, to this place, to this place, and to this place, and likewise. Now, after amplification, what do? You try to run a gel. Let us say we are trying to run a gel. Now, see, when I'm talking about gels in mini satellite, microsatellite, and AFLP, you are only running page. You're not running agarose. Remember that. Because to identify minor differences between the fragments, it's always page that identifies, and it is not agarose at all. Okay. Now in individual 1, because of the variation, and in individual 2, because of the variation, you remember this particular fragment is not cut at all in individual 2. You don't have a fragment here just because of the first reason that I mentioned, for polymorphism, presence of RE site. If it is not there, and if at all this is 500 base pairs, what's happening? You have a band here, but you will not be having a band here. You understand? There will be no band present here in this particular individual. Now, let us go for the second band. If this is around 700 base pairs or 750 base pairs, now you have a 750 base pair band here in individual 1. And here, let us say, just because of the presence of the insertion or deletion, if there is a deletion, what happens? The 750 will come down, and let us say there's a 400 base pair deletion. Instead of getting a band over here, you'll be getting a band at a 350 level because there's a 400 base pair deletion. And this is how polymorphism is identified. And the third reason I told you, presence of mini satellite and microsatellite. And the fourth reason is the binding of the primers because of the random triplet that is present at the 3. See, RFLP, PCR RFLP, yesterday we discussed. This is a hybridization technique, I told you, right? Because the probe is hybridized. And we have discussed about the types of probes here, the cDNA probes, and for the functional genes alone. And requirement of radioactivity, we said yes. And there's limited coverage because you're only targeting the genes here. There is a limited coverage of the genome. And yesterday we remembered this, you remember, right? We talked about this being single locus diallelic, and this is codominant. Similarly, you have a PCR RFLP here. This again, sequence specific primers involves PCR. Sometimes it is yes or no. You can also use radioactivity here, and it is limited to coding sequences, right? As you know, RFLP is always limited to coding sequences, single locus, diallelic, codominant. And possibility of automation, yes. This RAPD, arbitrary design of primers, that's what I told you. Yes, there is extensive genomic coverage. This is multi-locus diallelic we discussed yesterday. This is a dominant marker. And this is, again, what? Possibility of automation. Microsatellites, this involves PCR, I told you, right? Microsatellites involves PCR. When I said DFP here, it is nothing but what? Mini satellite. DNA fingerprinting. It is mini satellite. This involves hybridization. This involves PCR. This is sequence specific. This is mini satellite probe again. Involves radioactivity. Sometimes yes or no for microsatellites. Extensive coverage in both the cases. This is single locus because PCR is involved, right? And this is multi-locus, and this is codominant. I told you, explained clearly how it is codominant. Whereas here it is again, it can be automated. However, this cannot be automated. This is codominant. AFLP again, this is primers are adapter specific, I told you. Yes or no. Extensive usage. This is again multi-locus, right? Diallelic. And whereas here you have a dominant marker. Why is it dominant? Because initially when you are cutting your DNA with your fragments, like cutting your DNA with your restriction enzymes, this entirely depends on the presence or absence of your restriction site. Like presence or absence. If the restriction site is there, then you have a fragment. If the restriction site is absent, you don't have the fragment. So that's why this is said to be dominant, right? And one last thing before we leave today, right? We'll just get into this. One last thing that I wanted to discuss is, yes, we'll write the names of the persons who actually discovered these. The first one, right, as we remembered, the first one is RFLP. It is Grotziger. It is Grotziger in the year 1974. Right? Second one, RAPD. Because RAPD, it is Williams. Williams, 1990. Third one, mini satellite. Mini satellite, who's the guy? I told you, Jeffrey.
Jeff.
Yeah, Jeffrey, 1985. Also, there's a guy called as Nakamura in 1987. What else we have? 4 is microsatellite, Beckman and Weber in 1992. Beckman and Weber. 1992. And finally, we have AFLP by Vossital. AFLP by Vossital. 1995. Okay, guys. There ends the entire thing, and we have around 3 parts for